Good afternoon, everyone. Am I audible to you?
Yes, ma'am.
Okay. Shall we start now? Okay. Okay. So, good afternoon, everyone. My name is Shubhi Singh, and we will be having a lecture of um, thermal properties of food and rheology of food. So, starting with the thermal properties, uh, we will be presenting over the topic of differential scanning calorimetry. Okay. So, introduction to the thermal analysis. DSC, or the differential scanning calorimetry, is a thermal analysis technique used to study how food materials behave when they are heated or cooled. It helps us understand properties such as melting, crystallization, denaturation, and glass transition, all of which are very much important for our food stability and quality. So, like, we all are having different matrix of food. Okay, whenever we are giving any temperature, heat induced temperature or any changes to the food, what different uh, qualities, what different changes in the qualities are we are getting, that we are analyzing in the differential scanning calorimetry. It is solely dependent on temperature. As the name suggests, it is solely dependent on the temperature. Okay. So, thermal analysis is the study of material. I'll start the pointer. Thermal analysis is the study of material properties as they change with temperature. When any food is subjected to temperature high or the lower temperature, what different changes are inducing in the food matrix, we are getting the data and we will analyze it. So its importance is to determine food stability and the different transitions. Different types of thermal analyses are DSC, TGA, DTA, DMA. What are these? DSC is basically differential scanning calorimetry or calorimetry. Then TGA. TGA is a thermogravimetric analysis. DTA is differential thermal analysis. And DSC, we know, differential scanning calorimetry. So these are different types of thermal analysis. Among these, DSC, which is the most important, which is used for the food properties or food applications. Now, what is DSC? DSC is a technique that measures the difference in the amount of heat required to increase the temperature of a sample and a reference. The difference occurs because the sample may undergo physical or chemical changes like melting or crystallization that requires or release energy. For example, if we have subjected our food to heat, then it will absorb energy, and it will absorb the energy and it will get hot. And when if it will cool down, it will release the energy. So basically, this temperature change, AC always talks about this change of energy, this change of temperature uh, what is going through the food, what is happening inside the food matrix. This is what DSC is. So it is a thermoanalytical technique that measures heat flow associated with the transition in materials. Okay. Compare sample versus reference under controlled temperature program. So for reference, we can use water or any other solvent under which we have put our food matrix. The control temperature, under the control temperature program, we have to set a temperature range. Whenever we are doing DSC, we have to set a certain range of the temperature through which our food will be subjected to that temperature range. So we have to be make sure, and we have to take proper care which range of temperature we are choosing to, so that we can get the proper graph, and we can get the proper data of about glass transition or crystallization temperature, etc., which we will be discussing in the next slides. Principle of DSC, the basic principle of DSC is that when the sample experiences a thermal event, there is a difference in heat flow between it and the reference material. Reference material, which I have already talked, is we can take as a water, and our sample will be the food matrix, whatever you want to put, whether it could be a starch matrix or it could be a protein sample, anything you can use. So the basic principle of DSC is that when the sample experiences a thermal event, there is a difference in heat flow between it and the reference material. The instrument detects this difference and record it as a peak on the thermogram. What the graph which DSC gives is known as thermogram. Thermogram. Okay. So the graph will be like this, where heat capacity Cp will be here versus time. We can get a graph like this, then this, and there will be a decline. So this peak will be known as TG, or the glass transition temperature. So we will be discussing in the next slides. What happened basically, exothermic and endothermic, we all know exothermic reaction is the reaction where heat is released, and endothermic reaction is the reaction where heat is absorbed. So endothermic peaks correspond to the energy absorption like melting or denaturation, whereas exothermic peaks indicate energy release like crystallization or oxidation. Moving to the next slide. There are different basic components of DSC, like differential scanning, calorimetry. It has some basic components such as sample and reference pans. Pans are the ones in which we have to put our sample. There is a small pan. It is made up of alloy, and it is very much heat stable. It's not heat sensitive. It's heat stable pan, because whenever you are putting your food inside this pan, this metal or this alloy shouldn't be getting melted. It has to be stable. This pan has to be stable under extreme heat conditions. Okay. So there are sample and reference pan, then there is a furnace system, temperature controller, and data acquisition system. Here is a diagram of DSC. So sample pan, which I was talking about, sample pan, this is the sample pan. Then polymer sample, the polymer or any food sample you can take. This is a sample. Then there is a reference pan in which you are adding any other reference. Not this won't be included your sample. It will be including your reference. And then there are different heaters are there, which will increase the temperature, this one and this one. Then there is a computer to monitor temperature and regulate the heat flow. And also this computer will depicting the data or the graph. So basically it is having four different uh, components. Now there are different terms such as heat flow and heating rate. So can anyone please tell me about what is heat flow? Like the basic, you might have heard about the heat flow or heating rate. I want this session as a open there, like you can discuss and you can give the answers in this session. So I want you to please involve. Hello, am I audible?
Yes, Shubhri,
Yes,
you are
ma'am,
right.
it's audible.
Okay, so please, like, can you, anyone please tell me about what is this heat flow and heating rate? You can message in the chat bot also. Okay, okay, let's discuss. So there is a graph which is, you can see here, heat flow versus Q by T, or like the it is the heat flow is basically uh, at a given temperature. It can tell about uh, the heat flow is going to be shown in units of heat. Q is supplied per unit mass per unit time. T, the heating rate is temperature increase T per unit time. Okay, so heat upon time, Q upon T is the heat flow. How much amount of heat or is flowing with respect to time? Okay, in DSC, we do is we provide heat, or we increase the heat, we increase the temperature with respect to time. Um, Suppose 5 degrees Celsius per 5 degrees Celsius per minute, or 10 degrees Celsius per minute. We always increase the temperature with respect to time. So this is known as heat flow. And then temperature increase is denoted by delta T upon time. So this is known as heating rate. Basically, whenever in any formula or in any term, if rate is there, then it means that it is with respect to time. This term is with respect to time. That's why rate is there. And heat flow is Q upon T, whereas heating rate delta T is upon heating rate is delta T upon time. Temperature increase upon time. And heat capacity is again heat flow divided by heating rate. Heat flow is again Q by T. I'll put the highlighted. Q upon T is heat flow, and delta T upon T is heating rate. So when you will divide these terms, you will get Cp, or heat capacity. So this is the basic terms. While we are dealing with DSC, we have to keep all these terms in mind. So there is another important term known as glass transition temperature. What does it mean? Glass transition temperature. Basically, we have heard in class 12th about amorphous and crystalline matrix, or amorphous phase or crystalline phase. Okay, so amorphous phase is what? Amorphous, can anyone please tell what is amorphous and what is crystalline? Can anyone tell me the difference?
Amorphous means the structure is different or it is not, like heterogeneous kind of thing,
Yeah,
the
heterogeneous.
crystal structure.
Yeah, correct, correct. And crystalline? Crystalline is?
Uniform
Okay.
in structure.
Yeah, uniform. Yeah. Homogeneous or uniform. Like the interspatial voids, or the voids which are there in between the cells in crystalline, it will be uniform. It will be equally distributed. While in case of amorphous, it won't be equally distributed. It will be scattered. It won't form any uniformity. It will be scattered, and amorphous structures don't behave like homogeneous. They have uneven amount of gases or the voids present in them. So basically, glass transition temperature is the temperature when any food matrix or any polymer matrix loses its crystallinity. Crystallinity, when it loses its crystallinity, what does it means? Like whenever you are giving high temperature, under high temperature, the food matrix will start to melt, and when anything melts, it loses its uniformity, loses its crystallinity. We can take an easier example of ice. Ice is uniformly structured. It is in the form of cube. But when you melt it, what happens? When you melt the ice loses its crystallinity, and then it converts into water, and water is again a amorphous structure. Amorphous means it's not having any definite shape. It is not having any definite volume. So glass transition temperature is the temperature when your sample loses its crystallinity. So here, heat flow, with the respect of heat flow, like if you will increase the heat, okay, what will happen? Your sample will lose its crystallinity, and there will be a deformation in the graph peak, and then uh, different like it will lose its crystallinity, and we can record this as a glass transition temperature. So this is very much important term when we are dealing with when we are dealing with DSC. Also, we can say like we have also got an increase in the heat capacity of our polymer or the food matrix. This happens because the food has just gone through the glass transition. And as you learned on the glass transition temperature, food have a higher capacity above the glass transition temperature than they do below. Because of this change in heat capacity that occurs at the glass transition, we can use DSC to measure a food's glass transition temperature. And you also may notice that change doesn't occur suddenly, but takes place over a temperature range. Like if you are constantly giving higher temperature, it won't the glass transition temperature won't occur suddenly. It will occur after a some certain period of time. Okay, so when they reach that like over a temperature range, this makes picking one discrete TG kind of tricky, but we usually just take the middle of the incline to be as TG. Like there is a graph, and the moment when the graph will start having a peak. Then from there only we will record glass transition temperature. Okay. So there are different types of DSC instruments available for measuring food matrix, different types of food matrix. First one is heat flux DSC. It measures temperature difference between pan and difference between the sample and the reference and converts it into a heat flow signal. Okay. And there is the second one known as power compensated DSC. It maintains both pan at the same temperature by adjusting heat power. Heat flux DSC is simpler, while power compensated DSC offers better accuracy and faster response. So this one, the heat flux DSC, is considered better as compared to the power compensated DSC. So the working mechanism of DSC is, the procedure is quite straightforward. Okay, we take a small sample around 5 to 10 mg, and then we seal it with the aluminum pan. If you are taking a dry sample, I'm talking about dry sample, both dry and liquid sample you can do in DSC. But I'm telling right now we are dealing with dry sample. So for that, we take a small sample of around 5 to 10 mg. 5 to 10 mg is taken, and then we seal it with the help of aluminum pan, which we have already discussed. The DSC heats it at a constant rate, such as 10 degrees Celsius per minute or 5 degrees Celsius per minute also you can do, according to based on your literature. The instrument then records the difference in heat flow between the sample and the reference. We have already talked about the difference between the heat flow of the sample and the reference is measured by the DSC machine. Okay, whatever difference is there in between the sample and the reference, we record it in the form of thermogram or in the form of graph. The instrument then records the difference, and then from this we obtain a graph, the DSC thermogram, which shows thermal transition of the sample. Okay, record heat flow versus temperature and then analyze thermogram. Thermogram is the graph. It's nothing but the graph like this. Then how, after getting the thermogram, how we will interpret it? The x-axis will be the temperature and y-axis will be having heat flow. X-axis and y-axis. So x-axis is the temperature and then y-axis is the heat flow. Q by T, which we have studied before. This is heat flow. Okay. And then determine Tm, melting temperature, Tg and Tc values. We can determine what is the melting temperature, what is the glass transition temperature, and what is the crystallization temperature. So common thermal transitions in food are such as glass transition temperature, melting, crystallization, protein denaturation, and fat polymorphism. Protein denaturation is the temperature where your protein like alpha, beta, and different structures are there, and primary, secondary, tertiary, quaternary structures are there. And when those tertiary and quaternary structure loses or when they degrade, they eventually become denatured. So we can analyze our protein denaturation also with the help of DSC. Okay. So basically, glass transition is an amorphous carbohydrates. And carbohydrates we can see usually deal with glass transition temperature, melting in fats or crystalline sugars. Crystallization during storage we can see. Protein denaturation during heating, like if you are dealing with the milk or the paneer or the yogurt, milk based products, you can see the protein denaturation. Moreover, if you are dealing with the like if you have extracted protein from your pulses or any soya bean or pea, so you can make it into a powder, and then you can see and you can analyze how the temperature affects the protein denaturation in your sample. Okay, so this is the protein denaturation, and then fat polymorphism, which affects texture and appearance of food products like chocolate. Like if you had made a chocolate and you want to see whether like if temperature increase, what will happen when you will increase the temperature. So it also affects your fat structure and mouthfeel as well of your chocolate. So these different terms you can use, and you can see when while you are analyzing your sample in DSC. So sample preparation we have already seen, 5 to 10 mg sample is required. Seal the pan to avoid moisture loss, and uniform particle size and moisture content is essential. DSC in carbohydrate analysis, starch gelatinization and retrogradation. So basically, whenever we are using any carbohydrate or any starch sample, and we want to check whether at what temperature the starch loses its crystallinity, then we always deal with gelatinization and retrogradation. Whenever we are dealing with starch, we always see the its gelatinization and retrogradation. So can anyone tell me what is this gelatinization retrogradation? You might have studied. What is like whenever we are dealing with starch, we might have heard or we might have gone through these two terms, gelatinization and retrogradation. Can
Swelling of
anyone?
starch,
Yeah, swelling of starch is basically
gelatinization.
under which conditions? Swelling of starch under which conditions? What conditions?
When heat is produced, like mostly when we cook a starch, amylase
Yeah.
and amylopectin, there will be swelling in the starch as it gets cooked. Mostly when we cook a
Yeah.
rice.
I would like to correct you. When you are heating the starch, you have to give water also. Don't forget that. Heat with water. Then only it will get swelled up. So don't forget, whenever you are saying heat, you have to say water also. You have to provide moisture also. Then rest of the things are correct. So starch in the presence of heat and moisture swells up, and then eventually it bursts out, and then it forms a gel. It forms like a gel, which is known as gelatinization, if we talk in a easy language. Okay. And can anyone please tell me what is the temperature of gelatinization of starch? Like at what temperature starch gelatinization occurs?
Around 70, 72.
Yeah, 65 to 70 is the range where starch happens, like starch gelatinizes. Okay. And retrogradation.
Reassociation of starch molecules.
Yeah, reassociation of starch molecules when kept after so long, like after 1 to 2 hours. If you will keep that starch as it is, again this rearrangement of the molecules will happen. Okay. So in DSC, what happens is, like, we can see the starch gelatinization temperature and retrogradation also. While we are dealing with DSC in starch, we usually say the starch gelatinization temperature as instead of glass transition temperature. Okay. And sugar melting and caramelization we can see in DSC, and then impact of processing on carbohydrate functionality. Like if you are processing your starch, such as some people make like what kind of processing you are doing. So if you are, whether you are doing dry heating method, dry cooking, or whether you are doing wet cooking. Dry cooking, if you are doing, then Maillard reaction will happen. Okay. And if you are giving wet cooking, then gelatinization and retrogradation can happen. So it is subjected to the type of cooking method or type of processing method you are giving to your starch sample. Okay, so based on these factors, your graph, your thermogram will be made. Okay, or your thermogram will be affected based on these mentioned factors. Okay. And similarly, we have seen protein studies, protein denaturation temperature. While we are dealing with protein denaturation temperature, we will say protein denaturation temperature instead of glass transition temperature. Then enzyme stability, we can check the enzyme, whether our treatment, whatever treatment you are providing, whether your enzymes are stable or not. You can check with the help of DSC, and then you can see the texture and functionality correlations as well while we are dealing with DSC in proteins. And similarly, when you are doing DSC in lipid analysis, then you can see the melting and crystallization of fats. Okay, so fats are basically of two types: liquid and the dry one, the solid one. Solid ones are the ones which are solid at the room temperature, and liquid ones such as ghee or lard, they are the ones which are in liquid state. Okay, so you can check whether at what temperature, at what heat rate your fat starts to melt, or at what temperature your fat starts losing its crystallinity. Okay. And fat polymorphism also you can check. You can see such as alpha, beta dash, and beta forms are there. So you can check whether at what temperature this polymorphism is occurring, or your fats start losing its crystallinity. Influence on texture and shelf life. So all these analysis you can see, or all these results you can depict from the DSC of lipid sample. DSC is there, DSC in dairy and frozen food. Milk protein denaturation, again you can see, you can check. And dairy and frozen foods such as ice cream or frozen yogurt, also you can see different peaks and different temperature, at different temperature, how your food or your dairy and frozen foods is getting affected, and its stability in the frozen conditions. Applications in food processing, optimize heating and cooling process, ingredient compatibility, monitor structural transition during formulation. So basically optimize heating and cooling process. So while you are making a food, you are developing any food food material, so if you want to check at what temperature your food is started losing its crystallinity, or at what temperature if you are giving high temperature beyond this temperature, what will happen if you will give like more than, suppose you are making a peanut butter, okay, peanut butter, or you have incorporated some essential oils, or you have incorporated some essential oils, or you have added polyphenols into a peanut butter to prevent its rancidity, okay, and you want to check at which temperature my peanut butter will lose its properties, it will lose its structural integrity. So you can see with the help of DSC, DSC will help you to take to maintain an optimum temperature for your peanut butter, and then you can have and you can store it at that temperature only, that ambient temperature only, or the optimized temperature only. So DSC is very much helpful for predicting the storage life, shelf life, and the storage conditions of your food formulations. And hence DSC helps in quality control as well. It helps in detection of adulteration in oils and fat, shelf life prediction, and batch consistency testing. It helps to predict the shelf life of the food, and then it helps in adulteration, detecting the adulteration of oils or fats. With the help of DSC we can and hence with the help of DSC we can do the quality control as well. The advantages are very simple: small sample requirement, then high sensitivity and reproducibility, then it is having a quantitative results. We can as we have discussed, DSC provides a thermogram, and then thermogram also you can measure those results, and you can make a graph also, and then you can, it is quantitative results, like it is based on the data, it is based on numbers. So with the help of numbers you can easily analyze, you can easily get your results with the help of DSC. But it has certain limitations as well, as it is expensive. It requires trained operators. Anyone, like you, if you want to operate DSC, you have to first get a training, proper training, then only you can do it. And it is not ideal for volatile materials. Why? Because volatile materials are those which is like if you will have if you will add more temperature, or if you will increase the temperature, they will certain volatile components can be exposed out, and they can uh, diffuse out into the air. So the volatile materials you can't use for DSC. This is a limitation. These are the summary and references. Now we will see the video of DSC, so sample preparation and how do we operate DSC. Okay. Hello everyone. For DSC, we have to measure 1 gram sample of starch. Actually, here I have taken 1 gram, but for DSC we have already read that we need 5 mg to 10 mg 10 mg sample. Okay, but since this instrument is very much like it was deviating, so I had taken 1 gram sample, and then I mixed 100 ml of water. So accordingly you can measure and you have to be much precise, and you have to, like, while you are taking starch sample, you have to take it 1 to 2% concentration only, not more than that. Now there is a demonstration of DSC instrument which is available in our central research facility. Okay, so you can see the machine is, it's over here, and then there is a computer which is depicting, which is giving the results. Okay.
ये जो एपेटाफ है, इसमें एक में हमारा सैंपल है और एक में हमारा सॉल्वेंट है. अभी हम लोग इसमें रख रहे हैं ताकि हम लोग अपने सैंपल को डीगैस करेंगे. जो भी एयर बबल्स को एलिमिनेट करने के लिए हम लोग ये सब करते हैं, ताकि जब भी हम लोग डीएससी का रीडिंग दें तो उसमें कुछ अब्रप्ट पीक्स ना आ जाए. स्टार्ट हो गया ऐसे. अभी हम लोग इसको 30 मिनट्स के लिए डीगैस करेंगे. बच्चे लोग मैम को बैठने
ये in this whatever like we have to degas first while if you are using any start sample or any kind of sample you are using, you have to firstly degas the sample, then only you will have a sharp peaks and sharp curve of the in your thermogram. Okay. And there is sample loading, and they will depict how to there are different types of cells in which you will put your sample. See,
यहाँ पे हम लोग का दो सेल होता है जो कि यू शेप में होता है, इसीलिए हम लोग को चार दिख रहा है. एक रेफरेंस सेल होता है, यहाँ पे डालोगे तो यहाँ से निकलेगा, और एक सैंपल सेल यहाँ से डालते हैं तो यहाँ से निकलता है. ओके.
so basically there are 4 cells you can see, but it is in U shaped. Okay, so if this is a slot for reference and this is a slot for sample, you can put your sample reference from here, and it is in U shape, so it will reach until here, like it will reach till this level. Okay, and similarly for sample also, you will put your sample here, and then it will reach till this point. Okay, it is in U shape. So there are 2 slots basically, one for reference and one for sample.
जो कि यू शेप में होता है, इसीलिए हम लोग को चार दिख रहा है. एक रेफरेंस सेल होता है, यहाँ पे डालोगे तो यहाँ से निकलेगा, और एक सैंपल सेल यहाँ से डालते हैं तो यहाँ से निकलता है. ओके. यू शेप मतलब इसलिए क्योंकि मतलब इक्वली, मतलब जो भी सैंपल है ना, एक ही बार वो हीट हो जाए, एक ही बार में. इसको हम लोग लगाते हैं ताकि हम लोग टेम्परेचर सेट करते हैं, और ये टेम्परेचर 30 से 50 के अंदर रहना चाहिए. ओके, स्टैंडर्ड.
So this is talking, she is talking about the like after putting the sample, after putting your sample with the help of pipettes. Okay, you will close the lid, and then you have to set the temperature from this control panel. Okay, so here the temperature is set at 25 degrees Celsius. Then dp is in the form of calorie per minute. Okay, and then the pressure we have to fix as like around 5 to 10 psi. Okay.
Actually, यहाँ पे डाटा आपका डाल दिया, ठीक है, जो है, और इसमें क्या होता है ताकि हम लोग अपने कंपाउंड का स्टेबिलिटी चेक कर सकते हैं। तो इसमें करते हैं ताकि हम लोग अपने कंपाउंड का स्टेबिलिटी चेक कर सकते हैं। कंपाउंड का स्टेबिलिटी चेक करना, जैसे कि हम लोग इसमें हमारा टीएम दिखता है, बेसिकली हम लोग टीएम वैल्यू दिखता है। ये जो ग्रीन लाइन होता है, ये बेसिकली रिप्रेजेंट करता है आपका टीएम वैल्यू।
TM is basically temperature at which the sample will start to melting temperature.
मतलब जिस पॉइंट मतलब लिफ्ट होना स्टार्ट करता है। ओके, तो हम लोग ये टेम्परेचर रेंज लेते हैं और उसमें हम लोग को टीएम वैल्यू मिलता है। लेट्स सपोज हम लोग कुछ लाइगैंड वगैरह के कुछ अदर एडिटिव्स हम लोग डाल रहे हैं हमारे सैंपल में, तो वो टीएम वैल्यू चेंज हो रहा है, मतलब बढ़ भी सकता है कि कम हो सकता है। लेट्स सपोज अभी ये 50 पे आ रहा है और हम लोग का टीएम वैल्यू कुछ एडिटिव डालने के बाद वो 60 पे आया, मतलब दैट इज मोर इट प्रोवाइड्स मोर स्टेबिलिटी टू आवर कंपाउंड। अगर कम हुआ तो इट इज नॉट दैट स्टेबल। और हम लोग का ये डीएससी का मेनली हम लोग यूज कर सकते हैं, मतलब टेम्परेचर चेक करने के लिए। जैसे कि हम लोग कुछ ड्रग प्रिपेयर कर रहे हैं, तो हम लोग का ह्यूमन बॉडी में वो उतना टेम्परेचर विथस्टैंड कर सकता है कि नहीं, तो हम लोग इसमें चेक कर सकते हैं उसको।
So there is like DSC not only has its application in food. Okay, likewise this ma'am she told like if you are preparing any drug, okay, and you want to see whether it is stable after engulfing or after having it, whether it is stable or not inside your intestine. So DSC, with the help of DSC we can see that also. And like it has many applications. DSC has many applications, not only in food, but it has application in life sciences also. It has its application in polymer science as well, and it has its application in metallurgy also. So DSC is something which is not limited to the food application. It has different and it has wide range of application. Okay.
जब आप लोग मतलब रीडिंग ले रहे हो ना, तो आपको यहाँ पे ये मिलेगा कि टीएम वैल्यू कहाँ पे आ रहा है, ये सारा डेटा। बेसलाइन करेक्ट करोगे आप, जो आपने रेफरेंस किया था उसको बेसलाइन करेक्ट करोगे, फिर फिट करोगे। फिट का भी दो स्टेट मतलब डिफरेंट टाइप्स का हो सकता है, जैसे कि मैं यहाँ पे टू स्टेट ले रही हूँ। ओरिजिन
See, like
रेफरेंस किया था उसको बेसलाइन करेक्ट करोगे, फिर फिट करोगे। फिट का भी दो स्टेट मतलब डिफरेंट टाइप्स का हो सकता है, जैसे कि मैं यहाँ पे टू स्टेट ले रही हूँ।
the graph it is depicting the thermogram, it is in the form of CP versus temperature. So we can interpret the graph in two ways. Either this monitor will give the graph, or we can take the raw data and we can plot it into origin software. Okay, so it will provide different data, such as CP, and then heat or the heat capacity and the temperature. It will provide as a raw data, and you can plot it in origin software. But here in this system, it is already providing the data, so no need to plot it in the origin. it will give the graph as it is. You just have to do some baseline corrections in this software only. You don't need to install Origin for this machine for DSC. Okay, so sometimes you have to download, but in this particular system which we are demonstrating right now, for that you don't need to plot the data, like you don't need to plot it in Origin. It will automatically give the thermogram. Okay, see, this graph is there.
कि अगर आपका मतलब पॉसिबल हो सकता है कि आपके जो सैंपल में मोर देन 1 पीएम वैल्यू हो सकता है, तो ये भी यहाँ पे आ जाएगा। नहीं, मतलब इसी में ही हो जाता है, ये बेस लाइन में करेक्शन वाला है ना, इसी में करेक्शन। अगर आपका ग्राफ ऐसे आया मतलब ये एंडोथर्मिक रिएक्शन है, अगर डाउनवर्ड्स आया तो एक्सोथर्मिक, मतलब ऐसे पिक्स जो आया मतलब एंडोथर्मिक, अगर नीचे की तरफ से आया तो एक्सोथर्मिक।
See, the ma'am is talking about like we can get two different types of graphs, okay, in the thermogram. If the graph is increasing and then decreasing, then it uh, uh, denotes that it is endothermic reaction. Okay, and if the graph is in decreasing and then increasing, it means that it is exothermic reaction. Okay, so this was all about DSC demonstration. So this is the thermogram which we have got from the machine, from the DSC machine. This is a graph plotted between CP and temperature. See, we can see that this temperature around 73 degrees Celsius is the glass transition temperature. Okay, here the peak is of glass transition temperature and 73 degrees Celsius. Okay, so this is the graph, and here also we can see that this is this graph, this peak is 73, around 71 or 73 degrees Celsius. This is another graph for starch. It is measured in terms of in dP and temperature. This is the same graph only. Okay. So thank you everyone. And now we are open for question answers for DSC. After this session, we will start with rheology. If you have
Hello.
any doubt or any query, you can ask.
Hello, ma'am.
Yeah.
Ma'am, actually, for finding this heat capacity, I have given in DSC and I have received only the raw data. They told like we need to calculate it by ourselves. So can you please tell us how to calculate it using Origin for software?
Yeah, Origin, for this, as of now, I haven't made this for Origin raw data. It is the similarly, you can find, you can plot just like you have done for FTIR or in rheology, I will tell you, like it is the graph, it is the data you will find from the Excel sheet, and then from the Excel sheet only you can plot it in Origin. But in this particular instrument, the graph is already made, so we don't have to plot it in Origin separately. But the process is the same only. Like you will get the raw data and then you can plot it in Origin if you want to plot in Origin. Otherwise, no need of plotting this graph in Origin because this system provided the graph and we can directly use it like as it is. Okay.
Okay, ma'am. Actually, I have received the raw data only. That's why I asked. Like, I tried in the way day before yesterday or yesterday they showed how to use Origin software in this workshop only. But I
Yeah.
didn't get the baseline. I don't know how to get the baseline for analyzing. I just got the curve. Just and when we import the data in that software, I got the curve, but I don't know
Okay.
how to analyze it using. I don't know. I didn't get the baseline, and I don't know how to calculate also.
Okay, okay. So there is another method of, creating a baseline. You have to, like after receiving your raw data, you have to, create a baseline. And but as of now, we will only be like dealing with this DSC, and for this graph plotting, we can have another session, okay, where I will like we can have a separate demonstration of using Origin. Okay, so as of now, I can't open Origin and I will demonstrate you. But for that, we can have another separate session we will have, and then we can discuss that query.
Thank you.
Yeah. One question has come. How can we detect adulterants in oils and fat by using DSC? Is it based on the standard available data of oils and fats performance raised by? Okay. So of course, there are certain standards which are given by FSSAI. You know it very well. Like FSSAI provides different data, different standards regarding the oils and fats. They have categorized different fats and oils and different dairy and milk products as well. So based on those standards, we can easily determine whether our oil or ghee or the fat is adulterated or not. Okay. See, for example, chapter 2 of mm, chapter 2 of FSSAI. I can show you. Uh, I can show you in Google. Okay, so this chapter provides a detailed knowledge about different types of food products and what should be the standard. Okay, suppose for atta, for like it is for flour. Similarly, you can find for fat, oil, and emulsions also. There is another chapter for fat, oil, and emulsions. Wait. Mm, 2.2. Fats, oil, and emulsion. Yeah, this is the chapter where they have provided the standards for different oils, different fats, coconut oil. Okay, this should be the refractive index, this should be the saponification value, iodine value, Polinsky value, so and so on. Okay, so in this chapter, you can find the standards, and based on the standards, you can test your sample whether it is adulterated or not with the help of DSC. Okay, such a see, they have provided the standards for cotton seed oil, also groundnut oil, okay, मूंगफली का तेल, or flaxseed oil or linseed oil. So they have provided the standards. You can tally with, or you can like you can tally those results with your sample. Mahua oil, rapeseed oil, mustard oil. So different oils, they have provided the standards, and you can similarly tally or match your results, or DSC results with the oil of this FSSAI guidelines. Okay. So they have already provided the standards. Any more questions? So shall we start with rheology? Am I audible? Hello. Okay, okay. Okay, we can start with rheology. We have another session. Okay, we can start now. Okay. Is my screen visible?
Yes, your screen is visible.
Okay, okay, thank you. Wait, okay, so we will be starting the next session, rheology, rheological analysis of food. So rheology, the term invention, it has been coined by the Professor Bingham of Lafayette College. It is basically the study of the deformation and flow of matter. Okay. Rheologically basically helps us to understand the mechanical behavior of everything, like from toothpaste and ketchup to polymers and biological fluids. Okay, so if any kind of polymer or any kind of food material you have made and you want to uh, check the rheological properties, like its behavior, mechanical behavior. Mechanical behavior is the term which is very, very much important in terms of rheology. So mechanical behavior of any food product you want to check, you can do with the help of rheology. Okay, so its official acceptance has been it has been officially accepted in the 1929 by American Society of Rheology, ASR. Its scope and early applications, like it's not only it is not only dealing with the food. Apart from food, it has many applications, and it has been widely tested and tried for different fields, such as for lubricants, for paints, plastics, rubber, asphalt, etc. Okay, it has wide scope of subject apart from food. Modern advances and importances, the scope of rheology is now even wider. Significant advances have been made in biorheology, polymer rheology, and suspension rheology. So these are the different fields, different areas of research where the scientists have tried, and they are even trying, and they are even studying, doing research in these specifically mentioned fields: biorheology, polymer rheology, suspension rheology. It has crucial importance in the chemical processing industries as well, and future opportunities exist for extensive applications in biotechnological industries as well. Rheology, the word comes from Greek word. Okay, we can we can see it, the Greek word is known as Rio. Okay, Rio, RHEO. Rio. It is the Greek term, and which means flow. Rio means flow, and logi, or the logos, means to study. Means to study. Okay. So to be more precise, it is a study of flow of or mechanical behavior of the polymers or the food matrix. So we can see different types of liquids, such as non-ideal liquids. What are non-ideal liquids? Those are liquids in which the viscosity of a liquid may depend on the rate or the time over which shear force is applied. Shear rate dependent non-ideal behavior. There are certain liquids which behave as a non-ideal behavior. So what are those liquids? The viscosity of a liquid food may increase or decrease as the shear rate is increased, rather than staying constant for a Newtonian liquid. In these foods, the viscosity is referred to as an apparent viscosity, because it is no longer a constant. So those liquids in which if you are increase or decrease the shear rate, then they will behave as a Newtonian liquid. Some liquids are there which will behave as a Newtonian liquid, which will behave as a pseudoplastic fluid, shear thickening or shear thinning, or the shear thickening behave liquids. So they are all shear rate dependent. Like if you will increase or decrease the shear rate, they will behave differently. Okay. So in these types of food, the viscosity is referred to as an apparent viscosity, because it is no longer a constant, especially in Newtonian liquid. The two most common types of shear rate dependent non-ideal liquids are pseudoplastic fluids and dilatant fluids. Pseudoplastic are also known as shear thinning, and shear thickening is dilatant fluids. Pseudoplasticity and dilatancy are independent properties. Pseudoplastics and dilatant are time independent. Okay, you have to write it, or you have to see it more important. This is much more important, that there are two types of behavior: time dependent and time independent. So pseudoplastic and dilatant fluids are time independent liquids. What is shear thinning? Shear thinning is a tendency of some materials to decrease in viscosity when driven to flow at higher shear rates, such as by higher pressure drops. Okay, so when you are decreasing, like those materials which tend to decrease in their viscosity when driven to high flow at high when driven to flow at higher shear rates. If you are providing higher shear rate, if you are increasing the shear rate, like here it is shear rate, if you will increase the shear rate, they will decrease in viscosity. Shear rate, if you will increase here, then the viscosity will be decreased. So those fluids which whose viscosity decreases on the application of higher shear rate, those fluid are known as shear thinning. Okay, so the particles are like this. Wait, is my screen visible?
Yes, your screen.
Okay, okay. So with the application of higher shear rate, okay, with the application of higher shear rate, the viscosity will decrease. Those liquids are known as shear thinning liquids. Here you can see the molecules are firstly aligned, but when you are increasing the shear rate, they will decrease in their viscosity. And what are shear thickening fluids? Shear thickening are those which have tendency of some materials to increase in viscosity when driven to high shear rates. Okay, when you are increasing like you are enhancing the shear rates, their viscosity will tend to increase. So those fluids which viscosity will increase, which whose viscosity will tend to increase on the application of shear rate, those fluids are known as shear thickening. And those whose viscosity will decrease on the application of shear rate, those fluids are known as shear thinning liquids. Okay, so you can see in the diagram as well, equilibrium, like initially all the molecules are at equilibrium state, but shear thinning liquids are aligning more, and they are tend of making more they are aligning more, but whereas in shear thickening they are more dispersed, more dissipated. The molecules are more scattered in shear thickening. So these are two different types of liquid. So time-dependent non-ideal behavior. There are many foods whose rheological properties do depend on the duration of the applied shear. Okay, so shear rate you are applying, but for how shear you are applying, but for how much time you are applying that shear. So there are many different liquids whose rheological properties always change on the application of shear rate, like for how much time you are applying shear. Okay, foods such as suspensions, emulsions, and foams are time-dependent fluids and show thixotropy and rheopectic behavior. So, important examples of thixotropic and rheopectic are suspensions, emulsion, and foams, like are the time-dependent fluids. Those are basically emulsions and suspensions are basically time-dependent fluids, which show thixotropy and rheopectic behavior. What are thixotropic and rheopectic? Thixotropic fluid is one in which the apparent viscosity decreases with time when the fluid is subjected to high constant shear rate. Thixotropic is a term which is used for those liquids whose apparent viscosity will decrease with respect to time, or with respect to time when you are providing higher shear rate. Okay. The reduction in structural strength during the shear load phase and the more more or less rapid but complete structural regeneration during the subsequent rest phase. So after the applying the shear rate, the structure will regenerate, and it will make like the particles, the molecules will realign themselves, and those liquids are known as thixotropic liquids. Okay. We have already seen the examples of thixotropic liquids such as suspensions, emulsions, and foams. And what are rheopectic fluid? The apparent viscosity of the fluid increases with time when it is subjected to a constant shear rate. Okay, so in thixotropic, the viscosity was decreasing with respect to time, whereas in rheopectic, the apparent viscosity of the fluid will increase with respect to time when it is subjected to a constant shear rate. An increase in structural strength during the load phase and more or less rapid but complete decomposition of increased structural strength during the subsequent period of rest. Okay. In thixotropic, what was happening? There was a structural realignment, but here in rheopectic, there will be more increase in structural strength during the load phase, but complete decomposition of the increased structural strength during the subsequent period of rest. It will take more time to get realigned as compared to thixotropic. So this thixotropic fluid displays a decrease in viscosity over time at constant shear rate. The shear thinning fluid displays decreasing viscosity with increasing shear rate. Some fluids are anti-thixotropic. Anti-thixotropic means those who don't behave like thixotropic. So thixotropic we already have discussed is those fluid which whose viscosity will decrease over a time. So some fluids are there which are having anti-thixotropic material, which are behaving like anti-thixotropic, such as they are having constant shear stress for a time, causes a increase in viscosity or even solidification. See, so they even make a solid. They won't be stable. They won't remain in a liquid phase. They will make a solid material. Constant shear stress can be applied by shaking or mixing. How will, like, you will provide a shear stress? The stress you can provide with the help of agitation, or you can apply with the help of mixing or shaking. So shear on the application of constant shear stress, how your liquid is behaving, we can identify that. Fluid which exhibits this property are usually called rheopectic. They are much less common. So rheopectic are less common, but the thixotropic are more common. And there are some liquids known as rheodestructive. In some fluids, the time-dependent rheological properties are irreversible. That is, once the shear force is removed, the system does not regain its initial rheological properties. Okay. Like after the application of force, external force, when you will remove that external force, they will not regain its initial rheological properties. So those liquids are known as rheodestructive. Rheodestructive means they have lost their rheological properties. They are unable to regain, or they are unable to come to their original shape, or they are unable to reorient themselves. Okay, so liquid foods that experience permanent change are called rheodestructive. Like I have mentioned, they will be unable to come to their original position or original alignment orientation. This type of behavior might occur when aggregated particles are permanently disrupted and do not reform with time. So we have seen in thixotropic or rheopective liquid, what was like we have seen, like after the uh, after the passage of time, they are able to come to their original position, original alignment or original orientation. But here in rheodestructive, they will they will not regain its initial rheological properties, and they will experience a permanent change. Okay, so and this is the difference between rheodestructive and thixotropic and rheopectic liquids. So classification of non-Newtonian liquids. In this slide, we can see the classification of non-Newtonian liquids. Non-Newtonian liquids are of two types: time dependent and time independent. Time dependent, we have seen rheopectic and thixotropic, but there are certain time independent liquids also, such as Herschel-Bulkley liquid, Bingham liquid, shear thinning, or the pseudoplastic or liquids. Okay, so basic curve or the most important curve, the graph for different type of time dependent fluids are, this is a graph for Herschel-Bulkley liquid. This is the graph for shear thinning. It will, with the help of, with the increase in shear rate, the shear stress will decrease for shear thinning behavior liquids. Whereas in Newtonian liquid, it will form a straight graph. So with the increase in shear rate, the shear stress will also increase. Okay, for Newtonian liquids. Whereas in shear thickening, with the help with the increase in shear rate, again the shear stress will increase. Shear thickening. Okay, but it unlike Newtonian is forming a straight line, but shear thickening, it will form a curve. And for Bingham liquids, with the help initially with the help of shear rate, it won't form any curve, it won't form any graph, but when you increase the shear stress, it will start like it will start getting deformed. So now we can see the sample preparation of rheological analysis. For rheological analysis, how to prepare the sample for rheological analysis. Good afternoon, everyone. Today's practical session is about the demonstration of rheology and analysis of rheological parameters of food sample. For that, we are going to prepare the sample. The sample is starch extracted from tapioca root. Around 2.5 grams of starch is slowly mixed with 47.5 ml of distilled water in a beaker. The whole solution is kept at temperature of 90 degrees Celsius for the gelatinization of starch. Now our sample is fully gelatinized. Prior to the analysis, we will keep the solution at room temperature for cooling and stabilization of starch molecules. Now the sample is ready for analysis. So this was the video for sample preparation of starch, tapioca starch, and then this is how you will prepare your sample for the analysis. So before starting the demonstration video, I want to show you there are different types of probes which are used for rheometer. The rheometer is a machine in which rheometers are used to determine the deformation and flow behavior of all kind of material. The two most common rheological test types are rotation and oscillation. So rotation is this where viscosity law, there is a formula, uh, viscosity eta in terms of Pascal second is equal to shear stress upon shear rate. Okay, shear stress is the tau and shear rate is the gamma. So shear stress in the unit is Pascal and shear rate the unit is uh, second inverse. Okay, so shear stress you are providing with the help of rotation. Rotation is provided to test the flow behavior and viscosity of liquid with controlled shear rate via rotation speed tau. You are providing a shear stress with the help of torque. Here in the diagram you can see torque is applied, and then there is a measuring bob which is put inside the sample or the container. That now here it is, the sample is there, and then this is the measuring cylinder, and speed you can calculate. And rotation, this was the rotation, and here it is the oscillation. As we have discussed, we can provide into the most common rheological test. We can types of rheological test are rotation and oscillation. So the first one was rotation and the second one is oscillation. Here what happens is you have to calculate it with the help of elasticity law, where G is the complex shear modulus. Again, it is in the unit is Pascal, and shear stress, G is shear stress upon shear strain. Okay. Now here what happens is there is a measuring cone. The sample is placed here. The area is greater, but the length of the sample or the measuring plate is small. Unlike this one, here you can add large amount of sample in this rotation type, but in the oscillation type you have to use limited amount of sample. Okay. So we will see the different types of probes. This is the canister. This is the part of a rheometer. This is known as canister, in which you will put your sample. These are different types of probes. Okay, this is the flatbed probe. This is used for measuring, or this is used for analyzing different types of cookies or cakes. Okay, this probe is used, but whereas this probe is used for measuring starch sample, which we will be seeing in the next video. And this is another probe which is used for, again, you can maybe use emulsions, or emulsions such as mayonnaise or um, tomato ketchup, etc. You can use for this type of probe. So there are different probes for different food samples. Okay, so now we are going to see the video demonstration of rheometer. I'll stop sharing for a while and I'll start. Wait. Now we are going to see the demonstration of N10 bar rheometer. The sample is put inside the canister. The probe is allowed to move downwards with the help of control panel, where it mixes with the start sample with water. This process is known as pre-shear. Here you have, like we have already discussed about the probe, that probe which is used for, this is the probe, okay? So we have to fix it, and here it is a detector, it is a detector, okay? So with the help of detector, you will just insert this probe inside this detector and it will get fixed. And then with the help of control panel, which is over here, and you have to put the start button. You have to press the start button, and then the probe will start moving inside the sample. And the canister, which we have discussed, canister is the slot, is the, or you can say it's kind of a long beaker in which you have to add your sample. The sample is put inside the canister. The probe is allowed to move downwards with the help of control panel, where it mixes with the start sample with water. This process is known as pre-shear. The lid is put tightly to prevent the spillage of the sample. The experiment is started by pressing the start button from the input panel, and simultaneously the name of the sample is fed from the control panel as starch. Once you have given the input of start, then the spindle or the probe will started moving and revolving at a certain rpm, okay? And that rpm also you can fix, you can change, or you can change this rpm. So for this time, for this experiment, particular experiment, we have set the rpm at 160. Okay, 160 rpm is the rotation we have set. The whole process has been divided into 5 slabs. First slab is the pre-shear process, which is set at 50 degrees Celsius for 160 rpm. The second process is divided for the temperature range of 50 to 95 degrees Celsius, where the temperature is increased from 50 to 95 degrees Celsius for 160 rpm for 450 seconds. The third process has been set for 95 degrees Celsius for 160 rpm for 300 seconds. The fourth process is the decline phase of the temperature. The temperature is declined from 95 degrees Celsius to 50 degrees Celsius for 160 rpm. And the last process is set at 50 degrees Celsius for 160 rpm for 1 minute. The graph is being plotted and it will be interpreted in the next video. Now, the probe is allowed to move in the upward direction from the control panel. the spindle or the probe is being removed and the starch cell is also being removed. This portion is known as starch cell. This is known as starch cell. Okay, so we can remove this as well. This white color portion, we can remove this as well. This is known as starch cell. The probe is already being removed. Now they will remove this starch cell also. Now with the canister. At the last, the data in the form of temperature, viscosity, time can be saved in a separate file. And at the last, the result data can be saved and the graph can be plotted in the respect of viscosity versus time. Thank you. Okay, so that was the demonstration part, demonstration of rheometer, and now we will see how to plot the graph into Origin software. Okay.
Good afternoon, everyone. As you have seen already the experimentation of rheology. So by an example, I will show you how to plot the curves and how to interpret the data of rheology. So this is how we get the data of rheology. We can export the data in Excel sheet. So this is the flow curve data of a millet starch. So this is flow curve data where we can see the shear rate, shear stress, and apparent viscosity. In another sheet, we can see the frequency sweep test results, where we can see the angular frequency G prime and G double prime, which is basically storage modulus and loss modulus. So first we will interpret the data of flow curve. So for that, we have to copy the data to Origin. So here I have one Origin book sheet. I will just copy the data. I just copy the data from Excel sheet to the Origin. Graphs. For that, I will choose this line and symbol. Here you can see this is in the y-axis, there is shear stress, and in the x-axis, you can see shear rate. So this is the plot of shear stress versus shear rate. Similarly, I will plot for this shear rate versus apparent viscosity. Now, individually, we will talk about both of these curves, how to analyze and how to interpret this data. So as you can see, this apparent viscosity graph. So this is nothing, just apparent viscosity is the ratio of shear stress to shear rate at a given point in the flow curve. The apparent viscosity is a concept used to describe the flow behavior of non-Newtonian fluids. It's not like Newtonian fluids such as air or water that have a constant viscosity regardless of shear rate. This is non-Newtonian fluid that exhibit exhibit a viscosity that changes with the shear rate. As you can see, when we are increasing the shear rate, so the apparent viscosity suddenly got reduced, and it's kept reducing until it's got the flat line. So it's showing that shear thinning fluid. So there are so many kind of fluids in this rheology or food systems, such like shear thinning, shear thickening, Bingham plastic, thixotropic, or rheobactic. But in this case, in the case of starch, we mostly get the shear thinning graphs. So it means that as we increase the shear rate, as we increase the shear, so what is happening? That it's breaking the structure of the starch gels, so and making it more flowable. It's start flowing in this between the plate of the rheometer. So that's why the apparent viscosity gets reduced and reduced until it's get the flat line. Now we will talk about some this shear stress versus shear rate. This is nothing. This can this graph you can take as a description of this apparent viscosity graph. So what's happening here? This is the force. This shear stress is just showing the force per unit area required to deform the fluid. As I've shown, the structure get deformed. So that's why the viscosity is getting reduced also. So the force per unit area required to deform the fluid, it is typically measured in Pascal. And the rate at which the fluid is deformed under shear stress, it is measured in time inverse. Here, second inverse is the unit for shear rate. So basically, the shear thinning fluid considered as this, just like, for example, ketchup and oil. So generally, ketchup will not come out of this sachet until we press it, until we give some shear. Then when we'll give the shear, then only it will come out of the sachet. And so which is showing that it start getting flowing. So that's how it is showing. Or this is the reason we do the rheology of the gels and all this emulsions and film forming solution, this kind of things. So now we will move to the frequency sheet. For that, I will make a new sheet. There we have the data of frequency sheet. I will just copy it. I will paste it here. Now to name it. So this is our angular frequency. And this is our G prime, which is also called storage modulus, and G double prime, that is called loss modulus. Now we will talk about this in brief. So I will first plot these two graphs separately. Just select this x-axis and control plus select this y-axis, and then you choose this line plus symbol graph, and here you have two graphs for G prime and G double prime. So generally, to understand it in a simple way, we generally change the scale of x-axis and y-axis to logarithmic graph. So we'll make it here and (0.1 to 10). Sorry, this is up to 100. We have taken up to 100. So when we apply it, you see this graph is now in logarithmic. So similarly, we will do for this y-axis. So for here, we will take from 100 to 1000. We'll make it logarithmic to 800 to 1000. Let's see how we're getting this graph. So that's how we'll get in this for this G prime graph. So now similarly we'll make for this G double prime. We'll make 0.1 to 100, not linear, we want logarithmic graph. For vertical, 100 to 1000, make it logarithmic, click OK. That's it. So now these are two graphs. So, so why we are doing frequency sweep and what it describes. So frequency sweep test involves applying an oscillatory shear deformation to a material and measuring its response over a range of frequencies. The test provides insight into the viscoelastic behavior of the material by evaluating both storage modulus and loss modulus. So how we can say that how it's showing that viscoelastic nature of this material. So generally the G prime is showing the elastic nature of the material, and G double prime, which is loss modulus, is showing the viscous nature of the material. As you can say, as we are increasing the frequency, you can see that the storage modulus, which is viscous nature of the material, is increasing. And here we see that for storage modulus, as we are increasing the frequency, the storage modulus, that is the elastic behavior of the material, is also increasing. So that happens so many times in food structures, for generally for starch and any kind of protein also, that structure is not breaking and structure is keep giving the synergistic effect. So in that case, what happens that both of these behavior kept increasing to a certain point. So for this case, we are taking a fingerprint region. You can say that 0.1 to 100 frequency we are taking, and if we would increase it to like 1000, 10,000, so it may be possible that it will break and it will start decreasing. Any of them can give any kind of the behavior later. But in this region, for from 0.1 to 100, so this is clearly increasing in both cases. So this is the synergistic effect of the starch on gelatinization. Now, what happens that a high storage modulus indicates that material behave more like solid and with greater ability to store elastic energy. That this means it has a significant stiffness and resistance to deformation. And a low storage modulus, you can say, it indicates that the material behaves more like a liquid with less ability to store elastic energy. This means it has less stiffness and it is more easily deformed. While on the other hand, what happens, a high loss modulus, here you can see, a high loss modulus indicates that the material behaves more like liquid with a greater ability to dissipate energy as heat. This means it has significant flow characteristics and less resistance to deformation. Whereas a low loss modulus indicates that the materials behaves more like a solid with less ability to dissipate energy as heat. This means that it has a greater resistance to deformation and less flow behavior. But there are some other thing, like we can also interpret from these two graphs. So let's say I'm adding one more column. I will set as, you can right click on that and you can set as x-axis. So now this is x1. Now this column is x2. If I will add one more column, now it will be y2. I will copy this x1 because for both of these cases, storage modulus and loss modulus, both are same. I will delete this class. Now you got this G double prime, paste it here. Now this is x1 y1 and this is x2 y2. Now I want to plot both of this data together in a single plot. So what I will do? I will select it. I will go to the plot, and here you can see a double Y option. So you can just click on it, and here you can see both of these graphs, both of this curve is now plotted in the same graph. Now we have the common angular frequency, and on the right side, on the red axis, you can see that loss modulus, that is G double prime. On the left side, on the black axis, you can see that storage modulus, that is our elastic modulus, or you can say that storage modulus. This red curve is for this loss modulus, and black for the elastic modulus or storage modulus. So again, I have to make it logarithmic. So again, 0.1 to 100 and apply on it. And for vertical, 100 to 1000 logarithmic, apply. Okay, so now for this axis, it got logarithmic. Similar thing I have to do for the right side of the axis, red axis. So this is 100 and 1000 logarithmic, apply it. Now you see both of the curves has been plotted in the same graph. As you can see that this loss modulus, this G double prime is increasing, kept increasing, both are increasing, but at a certain point you can see there both are the graphs are cutting. So what does it mean? Like up to here, the elastic modulus means the elastic modulus in the material is dominating in the, up to some certain frequency range. The elastic modulus is dominant, and the viscous nature of the viscous behavior of the material is not dominating. But after a certain point, you can just zoom it out, and you can just select this screen reader here, and when you will click on it somewhere, you see around x is equal to 36.69. So at this frequency, so it's actually cutting. Both the curves are cutting to each other, which means after this frequency, 36.6 frequency, the behavior of the material will change. After that, viscous nature will dominate over this elastic nature. So that's how we interpret the data. That's how we tell the rheological behavior of the material. There are a lot of other experiments also for the rheology, but for time being, this is enough for this and to understand how to interpret the data and how to represent in our research work, our presentations, and this is the like a final form of the results of the rheology. You can just keep this type of the graphs in your research papers and presentation and your PPT presentation posters everywhere. So that's all about the rheology. Thank you very much, everyone.
So I hope that you all are like you have gained some knowledge of how to plot the video in how to plot the graph in the origin software. So that was the graph data interpretation part, and we have already seen the theory part. So thank you everyone for being there. Any questions and queries are always welcome. So you can ask. You can ask if you have any queries or any question. I will try to give the answer. Or you can put your question in the comment section as well. That's all for today.
We have
Yeah.
one question from a participant. That is when to use rotational and oscillation. Maybe in the rheology. Yes.
Okay. So as we have seen in the previous slides, rotation, different type of um, mechanical force we can provide in rheometer for rotation when we are such as rotational and oscillatory. So rotational we have seen whenever we are using large amount of sample or sample more than uh, around 1 gram or 2 gram we can use for that rotation. And for oscillatory, if we are using the small sample, like for less than 1 gram, and it also dependent on the motion. Like if you want to get the rotatory motion and you want to stir your sample very well, so for that you can use rotatory motion. And whereas in oscillation, as the name suggests, it will not uh, mix your sample well. Rather, it will just provide a shear force or the shear force to the sample. It won't mix your sample. Likewise, we have seen in the previous video where the motion was rotatory. The motion was rotatory in that demo video. And for oscillatory, you can use for the small part of sample, and where you want to just get the idea of you don't want to mix your sample, you just want to get the idea of like how much shear rate you are providing, and then what is the different changes which are happening in the food matrix. And maybe an oscillatory maybe used can be used for gels or mayonnaise, whereas in rotatory motion you can use for starch, protein solution, etc. Okay, like we have seen in the slide, right? I'll show you again this part. Rotation, it tests the flow behavior and viscosity of liquid uh, with controlled shear rate and determination of shear stress, like this one. If you are providing torque, like you are moving the probe, you are moving the spindle, and you are using rotatory motion, okay? And equation you will be using is shear stress upon shear rate. Whereas in oscillatory, what will happen? Small amount of sample or the shear stress will be used. The small shear stress will be applied to the sample. It will test the deformation and flow behavior of viscoelastic liquids and solids. Okay, so it is basically used for liquids and viscoelastic liquids and solid, whereas rotatory motion is used for more viscous liquid, less viscous liquid. Sorry. Okay, so this is the difference. Any more doubts?
I think
Shall
we
we
do
end up
not.
this? What?
I think we do not have any
Okay,
more
okay.
questions. We can end the session.
Okay, so thank you everyone for listening and having me. And thank you everyone. Now we will end up this session. Thank you. You can leave the meeting now.